thanks for having this idea of bringing the centers, people who are connected, affiliated informally with the SFIS, which I really can't say that fast, but anyway, with the group together, and thanks all my fans for, no, thank you everybody. No, it's great to see people from libraries, students, etc. Here. So Dave said, why don't you come and talk about your work, or the center, or something at the intersection. So I'm going to talk about my work, but in a way that's connected with lots of things going on in our center, and lots of things that other people are doing. And I should say that I haven't been at ASU the longest. My husband, Rick Creeth, has been here 7 years longer than I have, so he's the one who started all of this. He recruited me, I recruited Dave and Dan, or helped recruit Dave and Dan, etc. So it's a team work. Okay, so if I stand here, am I right here, or is it okay? Okay. All right, so, you know, we have to have branding, and this isn't the right ASU branding. I'd probably be arrested by the branding police, but oh well. Sorry, Andrea. Okay, so I want to look at, I'm going to talk about CRISPR as kind of a target, but really look a little bit about history, philosophy of science, policy, bioethics, law, lots of other things that all sort of come together, and that our center includes. And so let's just look at CRISPR, and then I'm going to go zoom back into the history, looking at what's new, and then ending up with where are we now. So, you know, we all know about CRISPR, and we all just, you know, it trips off our tongues to say clustered, regularly interspaced, short palindromic repeats. And we remember what that stands for, I'm sure. It was actually interesting at a AAAS meeting, one of the lead scientists, somebody said, what's CRISPR stand for? You know, there are a lot of science journalists there. So science journalists, it wasn't the lead scientist, but one of the significant scientists. What's CRISPR? And she mumbled and couldn't quite get it out. But yeah, so it stands for something, and every one of those words is important. So it's worth thinking about, you know, why it's palindromic and why that matters, why it's short and why that matters, why the interspacedness matters, regularity matters, clusteredness matters, and repeats matter. Yeah, so think about that, but I'm not going to read at you. I'm just going to say, you know, here's this thing that we all hear about. So what I want to look at is examples of science in the public. And then go back and look at a bit of history and science, not so much in the public. Okay, so I'm going to point to some articles from New York Times. There are other newspapers, but New York Times is useful to look at as something that's read by a lot of people and does responsible reporting of real facts and things like that. So here was an article by Pollock in 19, I'm a historian, right? Yeah, 2014. And so not very long ago, 2014, saying, hey, you know, in the 1980s there was this thing that they noticed, this science, but at the time they said the biological significance of these sequences is not known. Sort of like the Watson and Crick paper. You know, the biological significance of this double helix thing is not known. And then now the significance is known and is set off a scientific frenzy. And so how does something get known? You know, who knows it? What does it mean to know it? That sort of thing are questions that we can think about. And for anything related to embryos or lots of biomedical research, it's good to go to NIH.gov, which is a relatively reliable source for most things. And so they describe what CRISPR, and in particular the CRISPR-Cas9 system is like. I'm not giving a talk about the science, and if I were going to do that, I would ask Christian to do it because he's our grad student and he does it so much better and makes it fun somehow. But anyway, what's important here is that there is this technology, there is a perceived scientific frenzy, there is an attempt to make clear exactly what the science is doing, and the emphasis on using this technology to turn off genes, to edit out genes, to get rid of deleterious genes. That's the language that's used quite regularly. And then along with the discussion of the science comes from the beginning pretty much, okay, wait, but how we thought about this. Do we know how to control the results? We think we ought to need to do that somehow. And so that's a discussion that's there. You get pictures like this. So again, I'm not going to say, oh, and then the transcription. Though I know Victoria would love to hear about the science, but the transcription does this and blah blah blah. But there are pictures like this and cartoons and attempt to present the science to a public, to interpret it, or to many publics, and to interpret it in ways that are accessible. So okay, so CRISPR, excitement. And on the excitement side is, yay, it's great. It's a new tool. It can edit genomes, all good things, just like all those other words. So it can eliminate bad genes. There's a lot of language like that. Eliminating deleterious, eliminating bad genes. The discussion of gene drives is related, using a CRISPR technology to develop gene drives to drive through a population and edit bad genes out of a population by, in effect, editing good genes in. So the technology is emphasizing the taking out of the bad, and we'll see why in a second, very briefly. But versus this language right from the beginning is there. Oh, it's another version of a new eugenics. It's playing God. Oh dear, it's another technique that can allow us to create designer babies. There's a concern with gene drives, that we're driving something, we're going to lose control of it. It's going to affect the environment and populations in ways that are undesirable. So right away you've got this, oh, here are the bad things, here are the good things. And those are informed by discussion in the public of previous technologies. I want to say, but wait, it's not entirely new, so let's look back. So we can go a little way back and say, wait, there are other technologies that do almost the same thing that I'm sure you all heard about when they were developed, right? Zinc finger nuclease. Did everybody hear about that? Well, if you were in a biology department, yes. Or if you were paying attention, yes. But it wasn't front page news. Hey, there's zinc finger nuclease. And zinc fingers, I know that some people had vaguely heard of it, and I said, what do you know about zinc fingers? And they said, I don't really know. I thought copper is good for arthritis, but I don't know about zinc. So people didn't really get what that was about. But zinc finger nucleases are a technology that does basically the same kinds of things, being able to edit DNA sequences, edit out sequences that we want to edit out, or talons. That's another technology the same. So these three are all similar kinds of things, but with CRISPR, there are things that really are new. So it's the same kind of approach for the same kind of goals, but it really is a lot faster to use this technology. Zip, zip, zip. We can use CRISPR and zip out those bad genes just right away. It's easier to be reliable and to target specific areas, to say, okay, I'm going to target this area and be sure you've got it. So it's better, more successful in that respect. It's cheaper, much cheaper, because the other approaches take a number of different steps and checking things and culturing things and just more steps. And there's a sense that anybody can do it. And there was one report that some high school student who has really rich parents in the Virginia area outside Washington, D.C., but some high school student in his basement was doing CRISPR research. So anybody can do it, or lots of people can do it. Anyway, okay, so the idea, here's something that's great. It's going to be so much better than the previous things that were hard science and required a lot of knowledge and that sort of thing. So it will be, the idea is it will be more successful than any other deletion technologies, scissors type technologies that existed, and more effective and can do more things than traditional recombinant DNA research that we know about from the 1970s. So I know I'm kind of going quickly through this, but again, it's not a talk about the science, it's a talk about the science intersecting with policy, law, etc. So what's new, I would contend, with CRISPR, but also with a few other things. What's new is that we've had lots of science in the past. We've had zinc fingers, we've had talon research, but what we have is something that is very much in the public eye. And it's in the public eye in the context of thinking about genetic engineering or gene engineering. And so something has changed so that the science gets to the public more quickly. And so that's where we in biology and society like to say, and now for a little context. So we can all talk about the science, we can talk about the policy, let's talk about the history as well a little bit. So in history, let's go back 100 years ago or 125 years ago or so, a time when science seemed like a good thing and it was trusted. I mean, think about that. But, back when manipulating life seemed like a good thing, not something we want to say, what, manipulation? No, good, okay. It was assumed that we can be in control and are not gonna lose control. So let's sit back to the late 19th century and look at some old dead German white guys. So Wilhelm Roux, who there are a lot of more professional pictures, but I like this one where he looks kind of like a cowboy with his cowboy hat on almost. But so Wilhelm Roux and the next guy I'll talk about. But these guys, so in the 1880s, 1890s, they start doing experiments on embryos. And they talk to each other a lot. And sorry, Lexi, you heard me talk about this the other day. But they talk this part. But they talk to each other a lot, but not really in the public. I mean, there aren't newspaper reports saying, and Wilhelm Roux said, we need experimental manipulation. So Roux is doing experimentation. He said, let me take a frog egg. Let me see what will happen. I have a hypothesis that the frog's egg divides, so it divides into, one cell divides into 2, into 4, into 8, etc. That with each division, it's highly determined what each cell will become. So it has a fate, it's determined, he says. And so I have that hypothesis. I will test it by taking the 2-cell stage and killing one of the cells. And I predict that the other cell will do just what it normally would have, and the other is just inert mass. And so he says, see, I did the experiment, and it worked. I got that big blob of inert mass, but the other half is just what it would have been. Take his word for it if you're not a frog embryologist, but that is what it looks like. Okay, so, and he says epistemology is what matters. How we understand is what matters. It matters that we're experimental, that we're doing experimental work. And so that's the way we're going to gain knowledge about the universe, and this is a good thing. So again, it's not that there were newspaper reports, hey, half frog embryo develops into a half frog embryo, or epistemology matters, or let's do experiment. It just isn't in the public discussion yet. Scientists started to argue when this guy then said, I'm going to do a similar experiment with sea urchin eggs, which will be a better experiment. I can separate the 2 cells that are not stuck together. I can separate them. And he says, I predict that if Roux is right, I will get 2 half sea urchin larvae, sea urchin embryos. I predict I will get 2 halves. Lo and behold, he gets 2 smaller ones, but they're whole. And so it's different from Roux's result. And he says that's because there's some kind of vitalistic something, and metaphysics matters because what we have going on is something about the nature of the whole, the entelechy that's informing the whole. Never mind that whole theory, but the point is, here are 2 guys arguing about how to use experimental biology at the end of the 19th century in a way that the public does not care about. The scientists talk about it, they're all excited about it, who's right, Roux or Driesch. American scientists are very practical, they love their experiment, they kind of go with Roux. A lot of the Germans were entranced by Driesch's ideas. Driesch goes off and gives up biology and becomes a philosopher as a result of all of this. But the point is, it's not in the public eye yet. But pretty quickly it starts to be. So Jacques Loeb, working at the Marine Biological Laboratory on sea urchins, in 1892 discovers artificial parthenogenesis and talks about controlling life. So artificial parthenogenesis, he takes a sea urchin egg, he puts it accidentally, it turns out, but accidentally it gets into the wrong salt water concentration, partly because he was kind of a lazy experimenter and he just kind of somebody brought the sea urchin eggs and he dumped them in a bucket and it wasn't quite the same as ocean water, but that caused him to develop. So here is an egg and it hasn't been fertilized, so it starts to develop. And so that's pretty interesting. And so that reaches, that gets lots of publicity. First, this one, just in case you didn't notice it, but science nears the secret of life. Science nears the secret of life. So the science is now in the public eye. Theory of life, and this actually, it looks like it's an advertisement for the book, but it's actually at the start of a story that's about Jacques Loub's theory of life, mechanistic theory, conception of energy. So we're getting the scientific ideas into the public in certain ways. Tissue culture, Ross Harrison doing the first stem cell experiment. So he takes a neuroblast cell, he puts it in tissue, and cultures it. First time for stem cell, first time for tissue culture. And he says, look, the nerve fibers develop the way that they're supposed to. And so he said, he's a scientist, and so he said, he got funded and didn't have to work. Actually, he was a top-funded faculty member at Yale University at the time, and he didn't have to get NIH grants. So he didn't have to do translational work. So he didn't. He said, I did the science, I figured out the science, cool, I'm going to go and do more science. But other people picked it up and said, wow, that's amazing science, and so what can we do with it? So we have this ability to do tissue culture. I'm sorry this is chopping off the edges, but just imagine that the right words are in the right places at the edges. Yeah, so this was in the New York Times, 1910. Alexei Carrel at Rockefeller, explicitly building on Harrison's tissue culture work. We can build new tissue outside the body. So big headlines about that, and it was very exciting. Oh, we can engineer cells, we can engineer tissues, and this in 1910 was already exciting, but after World War I became even more exciting. So the science gets into the public discussion. Okay, Conch Bayman, another German guy, Bayman. So 1900 to 1930, Bayman working with frog embryos, does an experiment, and as I can't help but think every time how odd it is, takes the dorsal lip of the blastopore of one frog embryo, just the natural thing that you would want to do. Oh, I think I'll take the dorsal lip of the blastopore. He puts it inside, and lo and behold, he has induced another embryo. So he started with one and then induced a second. So he engineered so much that one became two, which is a pretty big engineering feat, and raised lots of questions. And so that got picked up also with a discussion about the Sculptor of Life. And then down here right below where you can see is it may lead eventually to man's being able to rejuvenate himself at will by putting the Sculptor of Life to work. So if we can know that it's the dorsal lip of the blastopore, or it's whatever, that you can just culture in a certain way, we can then culture what we need, and then we can solve all the problems, and we can rejuvenate ourselves. And rejuvenation and regeneration was a big topic at the time. So that also got talked about in the public. But notice something about the theme. Cool science. Wow, really great possible results, right? It's all really positive, and I've looked. It's not just that I went and cherry-picked or data mined and picked out the cool positive stuff. It's all positive. I can't find negatives yet. Okay, and so here, Beatrice Mintz, who takes two frog embryos and says, okay, I'm going to take two frog embryos. One's from a light color strain and one's a dark color strain. I can put those two whole embryos together. I can put them together and cause them, by a little nudging, I can cause them to snuggle up real cozy and nice and become one. So I can take two embryos. So before Bayman took one and made two, you can take two and make one. And it's normal. I mean, it's striped, but that's normal. It's a mouse, it's got all the parts. It's got the tail and all that stuff. So it works. Okay, so 1 + 1 = 1. So, the second is just taking a cell and putting it in a second. But here it's combining in ways, they're called chimeras. This work was done in the 1960s, and she continues to do related work. But, so it raises questions about embryos. So you might think, today, can you Well, try to imagine. So, what if this research were announced? Okay, somebody took 2 embryos and smushed them together and made 1. Maybe 2 human embryos. Oh, look, the IVF clinic got confused. They're not regulated after all. They got confused and 2 actually merged together and they touched each other and then they decided to join, but we didn't do anything about it. And then we get a perfectly normal little baby who's just maybe a little striped or maybe not, depending. But perfectly normal. I mean, it's a healthy baby. All parents want a healthy baby, right? If it's striped, it doesn't matter. Anyway, but okay, so what would we say? I mean, immediately that would be front page everywhere, right? Front page like, no, we can't do that. Play God, bad things, whatever. Okay, but here, multi-mice helped Dr. Mintz unravel some of the complex mysteries of cell differentiation. And then other stories go on. They enable her to do this and that. They enable her to study the effects of genes. We don't know enough about genes and their effects, so she can do this with this wonderful technology. So again, yay, science is good, science is cool, we can use it. All right, so positive in a sense, with science we can learn lots of things. We can learn things that we wouldn't learn otherwise. And so if we didn't do these kinds of experiments, we wouldn't have that knowledge. That knowledge can lead us to great applications, to good translational work. And so science is good. That's an implicit message really throughout, up until at least the 1970s. Then it starts to change. 1970s, recombinant DNA work. Recombinant DNA work with bacteria, viruses into bacteria. So who cares that much about bacteria? But people did because we're playing God. We're combining genes. Oh wait, genes are us. Genes are life. Genes define who we are. We're starting to mess around and mix them up. Oh dear, maybe that's not a good thing. I mean, before, okay, cool, good thing, interesting. But also in the 1970s, the recombinant DNA research hits the public eye right away. And so here's the science. Here's some scientists who go to Asilomar. Many of you know that story. But they go and they have meetings, and there are reporters there, and they report and they talk about this science, and they say recombinant DNA. And look at the headline: experiments with DNA have huge risks as well as benefits. Huge risks as well as benefits. So acknowledging benefits, it's not, all bad, run away. But okay, we've got to be careful. We've got to keep watch. We've got to keep watch on what's going on. And there are a lot of people who have talked about sort of why the tone changes in 1970s. I was at one meeting where somebody asserted, the 1970s. I mean, they were talking about recombinant DNA in particular, not just the way science is presented. But in the 1970s, there were bioethicists, and so the bioethicists screwed everything up. The one guy was contending. The bioethicists screwed everything up. Bioethicists started saying, wait, but maybe the science is bad. And then somebody else in the audience said, no, bioethics had existed before that. And then somebody else argued, no, it's not the bioethicists, it's philosophers, or it's reporters, or it's always somebody else. But there's a sense that things did change, and we're not sure, we collectively are not quite sure why. But DNA, there's a headline at the top here that says DNA and the public interest. So there's DNA in the public eye, DNA and the public interest. So what is the public interest? It's Stephen Toulmin article here. He talks about and asks, what is the public interest and how do we know? Okay, other technologies, so 1970s, sort of coming right after the recombinant DNA, in vitro fertilization. Not going to talk about that, you know what the basic idea was. In vitro, sorry, I shouldn't distract you. So with IVF, most of the headlines were healthy baby born and welcome Louise Brown, and then there were a few saying, oh, well, wait, what was behind that? And then in the U.S. there were a number of headlines saying, baby born in the UK with, from this technology, it was something like biotechnological baby. That wasn't quite right, but something like that. Born in the UK, can we do it here? So, oh, the US needs to catch up. That's probably a cool thing. So again, it was mostly positive, and that this could be a great thing for infertile couples. But there were some questions raised. Okay, but now we rush ahead. So I'm rushing ahead through a lot of things because I want to leave time for discussion as well. But so 1997. So I've zoomed ahead a couple decades, but yeah, we're getting to the point where you guys were born and know what was going on. So 1997, Gina Kolata, who this headline drives me crazy. Scientist reports first cloning ever of adult mammal. And then, it goes on in a feat that, is partly, genetic engineering that was anticipated and dreaded more than any other. So she sets up, some people are eager and some people were dreading it, and then she talks in the rest of the story about, they said it couldn't be done, and here it is, done in 1997. And so there's a tone of, not despair, but there's a tone of worry. Here's this technology, but watch out for it. And it's the first ever mammal, and mammals, a lot of people who read this, somebody did a poll, and a lot of people who read this didn't know that, humans and mammals are not coextensive. I mean, so that they thought that when they saw this headline that it meant humans had been cloned, and people, came to understand that, I hope. But anyway, so the way that this story was presented in the public, where Gina Kolata broke the embargo on the story that was going to come out in Nature, so it appeared in the press, in the public first, before it appeared in the science, and it gets framed in a certain way as a result of that. So that has consequences that are worth thinking about. And then in a year later, about stem cell research, scientists cultivate cells at the roots of human life. Anyway, there were a number of stories, obviously, about stem cell research, but the discussions about human embryonic stem cell research came out right after, just the year after, not quite a full year after, the cloning research was announced. And so people still have cloning in their minds. And so embryo research, oh dear, wait, we thought cloning was bad and it was presented as bad. Here's another one, maybe it's bad too. So it really colors the discussion. And people are not reading the scientific literature, they're reading, well, maybe the New York Times at best. Okay, so we have a case of science in the public interest. Okay, so science in the public interest, science in the public, means that it's science under scrutiny. And so we have these examples that we just talked about. Now let's come back to CRISPR and gene drives. So February 2017, National Academies report, which was labeled both by some of the people involved with it and by others as a new kind of report. A new kind of report that included governance. Now it's not, and Rachel knows quite well, it's not the case. This was the first time that any National Academies report ever said, and you should also think about governance, and you should also think about public engagement. But some of the people involved thought it was. They thought of themselves as doing something that was talking about the science, but also how we should govern the science. And so they present it that way. Sorry, and you saw from the title, Science, Ethics, and Governance. Okay, and it has 4 major recommendations, and so blah, 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 there are lots of words, but I highlighted the ones that are important. No, I highlighted a few. So there are 4 recommendations, 4 kinds of research. The first about basic lab work, which, we know what to do, fine, keep doing it. Somatic genome editing, yeah, we know what to do, keep doing it. And we should require broad public input. So that language appears. So before we extend the uses, so before we, not just the research, before we go and use this for a lot more things that we haven't done yet, let's have some broad public input, and we're going to require it. So it's kind of like all those words in CRISPR. Require, broad, public input. What does each of those words actually mean? And then third, germline genome editing, which is what freaks out especially bioethicists, but lots of other people, and maybe should give all of us pause, but we should even do research on germline editing only for very compelling purposes with stringent oversight, and we should have public participation. So public participation ongoing. Okay, and then with enhancement, where we're even going to go further and not just get rid of bad things and not just do some editing, but we're actually going to bring in new good stuff and enhance people. That we should really be careful about. Do not proceed at this time. Encourage public discussion and policy debate. Okay, so those are explicitly part of that report. Then copyright a year before, release slightly after the other report, but they're both going on at the same time. Committees met together a couple of times. Is this on gene drives on the horizon? You can't see it, but gene drives on the horizon. Advancing science, navigating uncertainty, and aligning research with public values. So there's what gene drives are. Doesn't matter in detail, but working on populations, driving populations is what's happening. And in the language of the report, engagement with communities, stakeholders, and publics is an essential part of research. So engagement, whatever that means, with communities, whoever they are, stakeholders, whichever ones we mean, and publics, whoever they are, is essential part of research and development of emerging technologies. But okay, so this is a strong claim that when you're doing research, you can't just do the research and then hand it off to people in SFIS and say, okay, you guys, now go talk about the policy. Or to somebody else and say, oh, go talk about regulation. It's an essential part of the research. And the question is not whether to engage community stakeholders and publics in decisions about gene drive technologies, but how best to do so. And so this is very significant language here. So coming to the end and moving toward discussion. So great, okay, engagement. Yeah, fine. What are the rules of engagement? You know, who are we engaging? Who's the we who are doing engaging? You know, so whether we should engage, why we should engage, how we should engage, who we should engage, to what end we should engage, what counts as successful engagement, and who count as publics and stakeholders and all of the other community categories. You know, those are questions. So how do we engage the public about science, and especially when the public doesn't trust, many people in the public, sorry, not everybody in the public trusts science. So what kind of engagement should go on here? And we don't really have very good precedents. We have some precedents, including some efforts with SFIS people. Yeah, okay, so maybe CRISPR-Cas9 isn't a new kind of thing, but you know, there's some sense that it could possibly have some new kind of effects, but at any rate, it arrives in a social context that really is different and is so uncertain about science and uncertain about what we mean by engagement that we have to think about what we're doing. So these are, I just pulled this from language related to CRISPR. What we need is engagement through anticipatory democracy. These terms in a number of places are not defined. I mean, I know people here are smart and you would all define them, but anticipatory democracy, okay. We think we know, we could imagine what that means. Participatory democracy, okay, we think we know what that means. Regulatory autocracy, that's what I read recently that somebody said, what we need is regulatory autocracy. We need the regulatory agencies to just tell us what to do. So all of this engagement stuff is they'll tell us what to do and then we'll go have a hearing about how we're gonna do it, kinda. Or, you know, do we need something else? So those are all questions. So how can you guys, now how can all of us, sorry, we the SFISers, you know, what can we do? Well, okay, so not this. So at AAAS, there was one point at which there was talk about engagement. They were doing a workshop, and usually AAAS, the American Association for Advancement of Science, is savvy and smart, and they know a lot about policy and how to work with the public and how to think about communicating science to the public in effective ways. But there was one workshop I went to a couple of years ago in which they said, well, you know, what we need is to educate the uninformed And then, we know what's ethically true and we know what's scientifically true. And so what we need is to educate the uninformed. And so engagement and communicating science means going out and telling people what the science is and then what's good about it. Okay, so not that. That's not what we should be doing, obviously. But there are things we can do. Identifying underlying assumptions is something that philosophers and historians do, as well as many others. But looking at what kinds of assumptions are behind the work that people are doing, noticing that there are different kinds of things that are counted as knowledge or that are counted as results in different cases, promoting conversations. So I wrote that down. I know that usually discourse or dialogues are the words, but okay, what does that mean? What does a conversation mean? But at least it sounds more neutral. So conversations where people are actually talking, and scientists and SFISers should engage and be willing to participate in public discussions. There are some cases where people have talked to me and said, well, I've called a dozen people at ASU and I can't find anybody who's willing to come and talk to my group of Jewish women, was one of them, or people in Sun City, or in different groups. And so, we have to be willing to engage in the public discussions if we think that they're a good thing, and provide a reflective approach. Okay, or you can write things. So this is just an advertisement, and most of you here have heard this before, but Slate Magazine has, feature magazine, I guess, Future Tense, which has short articles in it, and they welcome anybody to write those short articles. It's a collaborative venture with ASU and Slate and others. And so Bob Cook Deegan and I had one recently, and I want to mention this just because if you go to Slate Future Tense, you can find it easily. And these articles go up, and then people look at them for the next 36 minutes, and then a new one comes up. But what's interesting here is that we had right away 40-some comments, which is not very many, I think, but 40-some comments that were all positive. I mean, all of the comments were great. Okay, good. CRISPR editing, germline editing. If you can make people better, go for it. So interesting that the public discussion around other things is pretty negative, but here it was surprisingly positive. Now, that may just be who looks at Future Tense and Slate on a day when there's lots of other news and they can't stay away from CNN or Breitbart or whatever it is to see what he's doing. But yeah, nonetheless, okay, it's very positive. And so we say, and so Bob Cook Deegan really feels strongly about this last sentence here, but in the case of genome editing of human embryos, however, it seems likely technology will make the next move. So we can keep saying, let's have engagement, let's have discussion, let's promote public exchange of ideas, let's try to get ahead of the technology and the science in an informed way. But hey, look, technology is probably going to rush ahead. Now that technology doesn't rush by itself, but the science is going to get ahead of thinking about it. Okay, so yeah, so that's basically my end point. So let's have public discussion, but let's figure out what we mean. Let's don't expect that we're going to get too much agreement. Let's expect that it's going to be frustrating, even frustrating to figure out what we mean that we're doing and who we're doing with it, doing it with, and all that stuff. But a discussion about who we are and how we could do it well, where it is all that engagement stuff that the reports talk about.